Good evening. Welcome to everyone on what is definitely in Oklahoma, a cold, icy evening. And we certainly hope that those of you joining us this evening are somewhere warm with power. Obviously, you have internet access. It's certainly been disruptive in a number of ways, and we certainly appreciate your attendance this evening. I'm Jeff King. I'm on the executive committee of the DaVinci Institute. And here in Oklahoma, the DaVinci Institute is Oklahoma's think tank for creativity and education. And as part of what we do in that capacity, we hold a fall forum every year, which typically is a face-to-face event that's held on one of our member institutions' campuses. The pandemic, of course, is a disruption to that this year. And in the spirit of innovation and creativity, as well as knowing well the Zoom fatigue that has set in for everyone, it seems, in higher education, we decided that instead of our normal face-to-face mid-afternoon to or mid-morning to mid-afternoon event, we would hold a series of 4 1-hour Zoom broadcasts Monday through Thursday of this week. And so obviously we have had 2 so far. This is the 3rd in our series. And for those of you who are joining us this evening, we're absolutely delighted to have Doctors Donald French and Jeanette Louch with us for a focus on STEM education and STEM faculty in the age of disruption and the pandemic. And then tomorrow, our focus will be on liberal arts and education. And we're delighted to have Dr. Martha Parrott from Northeastern State University and a couple of her teacher ed candidates who will be with us tomorrow. And along with that, with her and her students will be Dr. Thomas Horn from Oklahoma City Community College, who will speak a little bit about teaching composition in the age of the pandemic and doing so at a distance successfully with some interesting technological approaches. And finally, Dr. Carrie Schneider from Cameron University will also speak and do some presentations related to the liberal arts. So just as a very brief preview of what's coming up tonight and then our final installment tomorrow night, I wanted to share that. And so before we begin with Dr. French, let me give you a very quick rundown of what our structure has been and the focus of what these what this series of 4 Zoom webinars is on and why we thought this would be an appropriate topic. The age of the pandemic has disrupted higher education enormously, but specifically for faculty, it has added any number of stressors. The pandemic itself is a stressor. The disruption of potentially being a faculty member with children at home because their schools are not functioning in a face-to-face mechanism is also a stressor. But teaching in new ways that may be a combination of face-to-face plus live streaming or fully online for the first time, having to function with the new pedagogy and or andragogy, as well as all of these other stressors, has really created a situation in which faculty can appreciate and can well use some ideas, some techniques, some tips for self-care and self-renewal. So we at the DaVinci Institute thought it would be very appropriate to try and focus around this idea of self-renewal and self-care, and to that end, share among ourselves, faculty in higher education in Oklahoma, in an informal setting, so that we can all learn some additional ways to help cope successfully in this new teaching learning environment. And so that's the reason for the focus of our 4-episode mini series, if you will. And with that then as a setting or a frame for this evening and the entire series, it's my pleasure at this point to introduce Dr. Donald French from Oklahoma State University, who is a professor of zoology there and the Edward Bartlett Professor of Pedagogy. Educated as an ethologist, he earned his PhD at Indiana University studying fish behavior, then joined the faculty at University of Maryland Eastern Shore, where his research focused on crab behavior. His interest turned to education and education research when he joined the faculty at Oklahoma State University. Since 1992, he's been coordinating the introductory biology course and has served as the coordinator for the university faculty preparation program. His scholarly focus has been primarily on implementing and evaluating evidence-based practices, active learning and inquiry-based or authentic research laboratories, for instance, in introductory courses, work that's been supported by HHMI, NSF, EPA, and OSU. He's served as president of the National Association of Biology Teachers and the Society for College Science Teachers, and has received awards for his teaching and or research from those organizations, as well as OSU and the Oklahoma State regions for higher education. So we welcome Dr. French to our program this evening, and we're so very grateful that you're able to join us, Dr. French.
My pleasure. So how would we like to start? There we go. Your slides are my slides.
If your screen sharing is working, you can do that.
Would you like me to do that?
That will work fine. That's worked well in our previous evenings.
Okay, then I will give me a half a second here to put some stuff away. I already hid my mail so my students wouldn't interrupt us with various things they do. And I will go ahead and share my screen. And let's see if that's working. We have that now? All righty. So thank you for the opportunity. That was a remarkably formal introduction for an informal setting. So I'm going to switch perhaps to something a little more informal. We'll see.
Please.
And what I'm going to talk about is delivering laboratory experiences remotely. That's been our biggest concern at the moment. I'm not sure. I think my contribution to our well-being is perhaps if we spend our time thinking about how we could plan it out, maybe there'll be a little less anxiety when we actually have to do it. So that's what I'm thinking at the moment. All right, so what I'm going to talk about is the planning. How might you plan a lab? Depends on what, as we'll talk about, depends on what your interests are, what your goals are. Some options that are available, again, depending upon your goals. And then finally, the approaches we've taken, which don't cover the broad range. Other faculty have, so I'm just going to talk about what we're doing specifically in the courses that I'm worrying about, concerned about. So the first thing really you have to think about is what is the role of the laboratory where you are, or for the course you have, and it could vary from lab to lab. One big one common thought often is that it's there to support the lecture. It's there to make sure that, you know, the students visualize or engage in the same kind of content that they might be seeing in lecture at that time. But you might have a whole different experience in mind. You might be focused on the process of science, discovery, getting involved in investigations. So there's a variety of things that you can think about to either expand or alter the content entirely or the focus entirely. Perhaps the experience you're thinking about is to provide an authentic experience. And that's, again, something that many people debate as to what does it mean to have an authentic experience, especially within a undergraduate course setting. And I know of a lot of ideas that people have, various places, including UCO, down at OU, et cetera, where people are trying to do this sort of thing. So when I say authentic, what I'd like to at least think about is these aspects, and this is a paper that was produced a few years ago, that it involves students in the use of scientific practices, all right? So the process of science, the kind of tools that we all use as scientists. It involves some kind of discovery. So there's something new to them. And then, and this is where a lot of debate occurs, how new does it need to be to call it authentic? And it's something we could all have an interesting discussion the rest of the evening or probably for days on end. It needs to be collaborative. Students are working together just like scientists do. Iterative, that is, it's not a one-shot deal. I don't just sit there, do this one experiment, and whatever I get for my results, that's it. They get an opportunity to work on a process, rethink it, reevaluate, and try again, and then broadly relevant to somebody. And this can apply in a wide range of fields. It's not just the sciences, but it should be relevant, hopefully, to someone besides just themselves or maybe the few students around them or themselves and the teacher. Who is it relevant to? So if you're looking at your own labs and thinking about this, you know, what are your expected outcomes? Is it content mastery? Are you interested in specific factual or conceptual knowledge? And that plays one role in doing a laboratory. And again, what I'm talking about, what doesn't matter whether it's online in our particular circumstances or face-to-face, we all have to face these kinds of questions. Is it about process mastery, design, interpretation, evaluation of data? How far do you want to go? Whose data are we talking about? What should people do with it? Is it skills mastery, analytical or technical? And there you might say, well, this, where we have a little more perhaps emphasis on the hands-on component of the laboratory as opposed to the design component of the laboratory. So again, it's going to depend on how you perceive the value of that particular laboratory, who your students are, etc. So there's lots of possibilities and combinations. Okay, so what's your approach? And I say this, we can talk about this in terms of inquiry, you can talk about this in terms of authenticity. And again, there are different viewpoints on that, but who sets the goals for the lab? Who chooses the topic? Who chooses the question to be investigated? Who chooses the materials, the designs and procedures, the analysis? Who's coming up with the conclusions? And this is a continuum. I see a lot of people over the years associate, for example, inquiry or authentic with something only on the far end, that it's discovery. The students have to do it all. Well, usually that's not the case. That's independent research. Maybe if you think about it, even doctoral students don't always decide what the question is that they're going to work in that laboratory. It often depends upon where the funding comes from, etc. So there's a range of things that can be done, and the teacher plays a different role in that laboratory and in making those decisions. So again, how comfortable are you? Where are your students? How well prepared are they? Can we get them, especially when we have to do things remotely, can we get them to whichever of these, to succeed in whichever of these tracks that we might perceive these as, okay, as to who's doing what, who's responsible for what. And to match those, there's a wide range of tools. And it's actually very interesting, having had a lot of discussions in the last few months on how to accomplish these. It's going to depend very much on what are the what the goals are, what your resources are, in order to decide. But perhaps if it's content, for example, then maybe if you're in an anatomy lab, use as an example, then images may be quite sufficient, videos. For some folks, it's about learning procedures, at least understanding what the procedure is about. And that, again, is being demonstrated by videos. We have people doing dissections live in front of a class when the animals are dead. But nonetheless, but the dissection. And they might be a video that's been done before. On the other hand, we have simulations, animations. We might move up to interactive lessons, case studies. If you've done work in the past, a lot of computer-based instruction has been a matter of setting up sequential environments in which students are asking questions, answering questions, and laboratories, even online, are getting far more elaborate. They often miss the goal of the iteration. They miss the variation in data. If you will, they don't fail as often if you look at a lot of laboratories, but that's not always true. But you can have, as indicated here, for example, my comment there about Ben's bad day, it's providing students with a scenario. They can work through these sorts of things, understand what the neurological, behavioral, in this case, processes, what's going on when this occurs. You see the little picture about the membrane potential. You can sit and you can adjust various parameters and get results that the students can then graph, work on, etc. And work from there. Databases and data sets. If you know anything about a lot of bioinformatics work, but there's other databases being developed, so students can work with that. And I'm going to give you a couple of examples of what we've been doing. But another approach altogether, which people say is, well, if the goal is to understand how science works, then working through primary literature may be quite valuable. Not just reading it once and talking about it, but basically deconstructing it, reconstructing, to decide what is the next experiment that might be done. How might this researcher approach this? And if there is a sequence of behavior papers, you could reveal them one at a time and say, okay, well, this is what you folks think. How would you collect that data? Let's take a look to see what the next step is. And in one of our other classes that I'm not going to talk about today, but well, just briefly, we do a lot of that. All right, where we sit there and look at and ask students to design experiments and then look at the results of others and then talk about what's occurring. So there's a wide range of things. And then finally, there's the idea of remote data collection. And there are sites around the country where some of that is available, where it can happen. A lot of citizen science is being done where remote data occurring. In our case, we'll talk about where there's someone in the laboratory doing work and the students are remotely collecting the data, or at least receiving the data, and then they can work on it. So they're missing the hands-on component, but they gain the rest of the experience. And I mentioned Merlot down here. I have some URLs at the end of the presentation, but Merlot is an organization. Oklahoma was an early participant, one of the founding members. Jim Bidlack down there at UCO still is extremely active. And this is an opportunity to have, so all of these items that I happen to pull up there, I did this on purpose, they were all pretty much pulled off Merlot. So there's opportunities to say, where do you get these resources as we talk about them to match what you're interested in doing. Okay. And finally, we might have to think about how do we assess, and I'm not going to spend a lot of time, but for identification of anatomy, maybe fixed response, maybe multiple choice would be a good idea. Maybe an essay drawing material will work up to that. For experiments that are longer term, any kind of work, reports or manuscripts. We have a little journal that we produce for our students, and they get the opportunity to actually publish in that journal. And I'll talk briefly about that. But poster sessions, which can be done also electronically, as we've been doing, is a well-known way of getting students actively involved. And it can be done online just as well as it can be done, perhaps a little less nervousness, depending on what it's like. It can be done online as well. So I'll talk a little bit specifically about our approach in doing this. We've been funded for a number of years now through a grant from the Howard Hughes Medical Institute for STEM persistence through authentic research opportunities. And we were providing these as both cures, so materials in laboratories, as well as mentored research opportunities through courses, as well as other events occurring to get students involved in research. But today I'm just going to really talk about a couple of things specifically with one of our labs, although we did do some work in other courses as well. And the course I'm going to talk about, not surprisingly, is the one that I coordinate. It's a mixed majors, so everyone's in it. There's about 1,600 over the year. As you can see here, they receive a topic, observations, a background. The students work in groups of 4, do the whole work of designing the experiment, conducting the experiment, and then ultimately they submit it to the Journal of Introductory Biology Investigations, where it's actually double-blind reviewed, and the students get back feedback and several times, so they get an opportunity to see how they can improve their manuscript. And hopefully at the end, there's 2 components. One, they do receive a grade based on it, but also the manuscripts that meet our criteria are actually published, and they have a citation, and they're rather proud of this, many of them, and this sort of thing. What do our experiments look like? We tried to make something authentic, connected up to our researchers at OSU. So one was on looking at maximizing microalgae growth. Picochlorum oklahoma was something that is local to Oklahoma. It was discovered by one of our faculty at OSU. The students use incredibly economical photobioreactors. That is, you see the water bottles that are there. Those are photobioreactors which the students can build and collect data on various parameters. So they can choose whatever they want to do to see if they can maximize this. And then they can learn the techniques of cell counts, of using mass and dividing to get an idea of how much the cells are growing. They can do some work on looking at characteristics of the cells, do chlorophyll counts, etc. So the chlorophyll measurements, so they can look to see what their parameters have been like. How successful are they in growing these better than has been done before? One of our other experiments, based on research that was done actually a long time ago, it was ironic. One of the other newer faculty came out of the same lab that I did when I did my PhD, and they use classical, ethological models where you present these to the fish, and you look to see what characteristics. I'm already in my fish mode. I put my hand automatically when I do this. We look to see what characteristics are attractive to potential mates. And the students are able to use these, but we have them design things with 3D models. All right, so those are how the labs were done traditionally. How did we do our shifting? Well, pre-COVID-19, the planning is done online. So we were in good shape there. They submit their plans for their experiments. They get their feedback before they come in. Experimentation was done in lab. The writing was all done in lab using Microsoft Office, and submission and grading also was done online. So they submitted to the journal where it was double-blind reviewed. So we had some things going for us that was well positioned for making a shift. So what do we have to do for a shift? Well, spring of 2020, you know, the panic we all faced. What do we do? And we had my TAs, there's a lot of them. They were outstanding in pitching in, and basically the students went remote and their hands were the TAs. And so their job was to give the TAs their specific directions. What do you do? What are you collecting for me? Now, the advantage was that depending what they were doing, once the experiments were set up, and you can see the TAs, if you see in the lower right-hand corner, there's a TA, she's actually talking to the students. So they had computers at every table, and the TAs would walk around the room just like they were walking and talking to each group. The other the students are online, and they literally are talking to them. What do you want me to do? They got a camera, they walk to the next table, they're talking to them there, and they're walking around the room. And with the remotes, with the software, the students could actually use Logger Pro, and they could see the data being collected as the experiments were going on, at least for the algae experiment and one of our ones on fermentation. They could actually see the data being collected. So it worked out nicely, except for the set of hands. Okay, well, that's great. How do we modify that for fall 2020? We have a different circumstance. So we went with many of the same things. The students could still do collaborative work using Google. They could work their documents. The TAs could watch them. They could see how they were working. And what we did was we brought half the team in. So 2 students in, 2 students out, so we could have social distancing. All right, a lot of work to set up the laboratory to make sure that nobody could breathe on anybody else. We're very careful, but nonetheless. But I want to make a comment about this, which is also interesting. We had an interesting problem, which was we have different combinations of students, some of which really didn't want to go back in the laboratory. Others really wanted to be in the laboratory. This is also true of our graduate students. Some of them were concerned about, how do I keep everything safe? And so basically over time, we said, okay, let's give them some freedom. And as long as someone's working, so for some TA group combinations, they had the 2 and 2. For some, they had 1 and 3. One student would come in and work in the lab, the 3 remote. And in some cases, they decided that the TA would be the hands and the students would be remote, but they're there on their monitors. The TAs can see them. We added document cameras so they can see the setup in front of them at all times. There's a camera on the person, there's a camera on the desktop. So they're really able to see all the experimentation going on. And we let students, we still don't know in the end how the different arrangements worked as far as final results, we'll see. But for now, we know that those laboratories are moving along. They're getting the full experience as best we could. Now, the fish lab was a little more complicated because it really takes direct observation. Setting up video cameras to actually get all the angles is difficult. So we went with the database approach. We have many years of data, which the students in the past have been collecting. And so we could have huge data sets of these model presentations, and the students can go in and they can look at the models and decide which model they want to use. And instead of just doing the normal comparisons, they could compare any set of models to any other set of models as long as they were tested against a common control. And we have lots of data like that, so we're able to move our students online entirely, and they're still working with very real data. Okay, so that's pretty much so it's a different approach there using our own data. And by the way, you can see our little 3D fish there. We built it so the students could come in and actually, whatever model they chose, they could bring it down and rotate it on its own screen so they could take the parameters and see exactly what their fish model looks like. And we're developing that for the future as well, so students can modify things. Okay, I'm going to take one other example. We have another course which is on DNA barcoding to characterize zooplankton communities. And this is really much more the true cure. It's based on Dr. Andy Jalowski's research. And you can see over here, this is some of the projects he's working on: detecting invasive species, restoration of fish habitat. And we're trying to develop techniques so they can effectively use DNA barcoding to characterize the plankton communities. So we have our students in. It is very much technique intensive: DNA extraction, PCR, gel electrophoresis. Some we can do some work. And so again, we paired the students so they could work and they could switch off which student is doing which work each week. And what I thought was worth doing is giving you a picture here of what the lab looks like. Here are our students. You can see at the moment they're learning how to use their analytical balances to get to take measurements. And we're actually watching them through the document cameras. We're remote. Here I am. I'm remote. One of the TAs is remote. The other one is in the laboratory. And if you'll see over here, there's a comment here by Jia Ren. She's a postdoc of mine. Unfortunately, she went to China when COVID hit, and she's been there ever since. So she's remote from China watching our students. So we're watching them all remotely. They're communicating with us over their microphones. We're watching the work they're doing. And even colleagues in China are giving them feedback saying, you might want to try this now. What should we try next? Critiquing their work and providing them with feedback. So I thought that was worth doing. We're very proud of this. This is our chat going on. And again, it's possible to do these sorts of things. Does this all work? Well, this is something we can debate, but at least we'll do the do no harm. Pre and post COVID, our grades, at least in the spring, there was no effect, no negative effect on this. This semester, it looks like some of our lab grades are actually a little higher. Now, we also have a little more disparity, and I'll be honest about some of this. We have some students that are not taking to, what I mean is they're not showing up when they should, and that presents a problem. And we have a way for them to work out, if they don't do the work, how they can compensate. But we are, I don't want to say losing, but we have some instances where we really have to reach out to them more and work on that, because we do have some that are not earning the credit because they're not completing the work they should. This happens throughout many courses. Let's not pretend, but we're just a little more aware of it under these circumstances. But the grades on the reports are actually a little higher than they've been so far, so we'll see. So with that, I thought I would leave, and maybe since we're supposed to have some discussion, a couple of questions we might want to kick around, depending on how you'd like to open the discussion. What are your goals? Okay, learning objectives for your labs. What do you value most when you think about this? And maybe we can talk about what might be a good approach. And then what challenges do you think you're facing? I mean, we're doing this with a large number of students and a small students. We're doing this with a lot of tech, and we're doing this with a lot of, some of our TAs are new to the campus, and some of ours are old hat and have done this numerous times before. So we've got a range of experiences. What might your challenges be? And with that in mind, I'll open that up for discussion, whatever you'd like to do, Jeff.
Thank you, Dr. French. Very fascinating how you made the transition, and all of us are well aware of the fast pandemic pivot, which I think is closely related to the funky chicken or the running man, but the pandemic pivot that was forced upon higher education around the time of spring break. The way I think we should proceed for just a few minutes here, there is one question already posted into chat for you. And while you're taking a second to read that, the attendees can also see that Mark Walvard has posted the link to Merlot. And please understand, whatever your discipline, Merlot is a wonderful resource which can provide case studies with video resources in support of the case studies, on pedagogy, on disciplinary content. If you haven't visited the Merlot site, please take a few minutes to kind of tool around. You'll be, I think, very, very impressed with the range of resources that you can put to use as a result of that visit. So with that, let me turn it back to you, Dr. French, for that first question.
Okay, so I'm taking them in some order. So one is this, how does this one's a fairly straightforward. How does this differ for your courses for majors? We believe wholeheartedly that right, the general education is we've made a course that's suitable for all students and prepares our majors. So we think that at this earliest level course, this is both majors and non-majors. And anyone that realizes that students are trying to decide what major they're going to be, plenty of our majors are not really thinking, they're not, I don't want to say they're not really majors, that's not fair, but they're entering it for a wide range of reasons. So the experience is good for everybody. They all need that sort of understanding of what science is. And once we started thinking about this way, how do you leave saying, I understand how to do an experiment, I understand how the problems that researchers face, the problems that science encounters, how to think this way. We're not sure if you said, should that have been for majors or non-majors to know what it's like to be to do science? And so that's what it's about. Plus, many of our students start out, what do we declare a major? Well, you can be any major and be pre-med. So what harm does it do? Okay. Another question here about, to make something authentic, they need to get publications, even undergraduate research journal or proceedings. So that is the interesting question about what does make something authentic. And there's a couple of different thoughts about this among people. The more inquiry-oriented folks say it is important that the students focus on coming up with design, making their own contributions, designing and taking ownership that way. And that discovery can be more local. There are others who say, yes, we want to contribute, and it should contribute to a far broader audience. And that can mean, so for example, when you say publication, if I'm doing, for example, what we're doing here with the DNA barcoding, those students are not necessarily getting publications, but the information goes to a database, or so any of our students, or it goes to the faculty member. It's data that's being contributed. So that's an aspect of authenticity. And it's interesting because if you think about a lot of research, the inquiry folks, if you will, put an emphasis on design. A different aspect of authentic say, well, it has to contribute to a broader audience. So one of the ways we dealt with this was creating that student journal. Mm-hmm. So every student has the opportunity to approach, to publish in that. And the journal is used by all the subsequent years of students. So they get to see the other research, they cite it, they work with it. Should they also cite other kinds of primary literature? Absolutely. But it is definitely the full experience in the sense of they are contributing to knowledge to multiple years of generations of students. So based on that, just like inquiry, there's a continuum of what is authentic. Hope that answers the question. Okay, anyone else?
Well, this is a beautiful segue into our comments from Dr. Jeanette Louch, and I'm hopeful, Dr. French, you can stay with us to the end, because I know that the connection between what you've just talked about and the things that Dr. Louch will share will mean that we may have a very robust combined Q&A at the end.
That would be fun. That was my plan, so I'll be delighted. Jeanette, I'm looking forward.
Wonderful. Well, at this point, it is my great pleasure to introduce to everyone Dr. Jeanette Louch. And Dr. Jeanette Louch's PhD is in pathology from St. Louis University. She's a molecular virologist and teaches introductory biology, genetics, microbiology, cell molecular biology, immunology, and virology. She mentors students in research and does STEM outreach for young women and men by way of introducing them to food science and food preservation. Dr. Louch has been at USAO since the fall of 2007. And so, Dr. Louch, thank you very much for being with us this evening and contributing. And once again, we will keep track of the questions that might come into chat, and we are happy to present you at this point.
Okay, thank you very much. So my experience is not quite as organized as Dr. French's experience. I actually had taken the summer off of teaching, my first summer off in 20 years. And so I actually took a bunch of classes to try to figure out what the best thing to do for online teaching actually would be in the laboratory situation. And so one of the things that I felt when we were having conversations about what to do with our students is I don't think that they were really considered much in the practice of what we were going to do in terms of the fall. So I actually didn't make a decision about my classes either being asynchronous or synchronous or in person or online until actually the first day of class when I actually met with our students. And I think Dr. French mentioned this too. Most of our students, or at least at USAO, a lot of our kids wanted to be in the classroom, which is all well and good, except when your laboratory has been deemed a 6-person lab instead of a 24-person lab. It made it quite interesting for me to come up with a way to teach. This semester, I'm actually teaching microbiology and its laboratory. and cell molecular biology, and then a new lab that we're actually trying to integrate biochemistry along with our cell molecular thing. And so we're making it a 2-credit course that meets twice a week so that the students can see a little bit more about what actually happens. And then the lab will actually cover the cell molecular lab as well as our biochemistry requirement lab. And so I'm trying to figure out, how am I going to take my 21 kids in cell and biochemistry techniques into a classroom that can hold 6? And how do I take my 18 kids in micro into a lab that holds 6? And so for me, I wanted that experience to be a hands-on experience. Our students in the past, in the 13 years that I've been at USAO, they take away a significant amount of tools from both, at that point it was the cell molecular biology lab and the micro lab, that has really enhanced their ability to get jobs. It's enhanced their ability to decide on graduate school or not graduate school. It's gotten them into summer research programs. It's introduced them to mentored researching, which we do here on campus. And so I couldn't, in my heart of hearts, let my students not have that same experience that lots of other students had in the past. And so one of the things that I was doing as I was getting ready to have our first meeting for the lab, since we couldn't all fit in the lab room, was to do a Zoom conference with them remotely. And as I was getting ready to start the Zoom conference, I got this like aha light shining moment, big ray of sunshine came down over my head. And I'm like, what if we ran this like a research laboratory? I actually gave them the opportunity to conduct experiments in small groups that all led to a bigger piece. We were going to have Zoom meetings in which we would talk about what they did, what the implications of what they did, and those kinds of things had on the overall impact of the experiment. And so truly, not only do we have this new class that's not been taught before, but now we have this very highly experimental method of teaching this particular class. And so we've gotten to where, we're still working out some of the bugs. There's a bunch of data I have to evaluate this week so that we can move on to the next step to determine whether our enzyme kinetics lab needs to have a few more tweaks to it, or if we're ready to start writing that up. So we meet about every 2 weeks as a group online during one of my hours of Zoom office hours. And we kind of talk about the things that we've actually been doing. And so I've broken up the groups in such a way that they only come like maybe once a week, or depending on if the lab is really short, we have an hour and a half group that comes at 3, and then we have a second group that comes at 4:30 about, and then they do their stuff, and when we kind of collect and make a data bank of the data and what have you. And so we're starting at that point where they're all getting the same types of laboratories. So this week, while we were supposed to finish it up on Friday, Western blot, looking at relatedness of some fish that one of my counterparts went out and caught at our habitat area. We have what used to be the old farm associated with USAO where campus was supposed to be built, that it didn't happen at that point back in 1908 or whenever that was. And so we've kind of turned it into a native prairie, and it's got some water. And so a friend, one of my counterpart teachers and his son went out and caught some fish. We're going to try to see, we're going to do some barcoding for the fish as well as this protein analysis to see if they have, proteins in common. Are those proteins in common enough to be recognized by the antibody? Are they the same size? So we can do, we can do protein determination. We can do, molecular sizing. They got the experience of running an acrylamide gel, a dry blot transfer, And now they'll get the opportunity to do the Western. Whereas the other group, they didn't get that same experience, but they got to set up an ELISA, and we made this story about who has COVID, who doesn't have COVID. And so we did a multi-step ELISA process so that they also get to see antibodies. And so when we get together this coming week, after we finish up the Western part of our protein study, those kids are going to explain the differences between those two techniques. And other than substrate, there really is not much, but it's a totally different process for them to get to see. And we are also kind of using that same sort of idea with our microbiology classes. They learned a bunch of techniques, like how to test for sugar production or sugar usage rather, and sulfur production and indole production. And now we're getting ready to give them their unknown final case study, because the majority of that class is actually kids who want to be physicians or they're nursing students. So they basically are all in the same 3,000 level micro class. And so they're getting ready to get big case studies. Each one of them will get a case study with a particular sample. So somebody will get salmonella, obviously, and it'll be in a Tootsie Roll. So we'll autoclave some Tootsie Rolls and inoculate that. And so they'll get very real looking samples along with patient data that I generate, because part of my previous world, I was a medical technologist. So I have lots of, gory microbacteria stories of things that I've seen in actual real life patients that I can then pull in and, give these particular things to students. And they have to be able to identify the organisms by the end of the semester. And so this idea of them sharing a little pieces of information and then doing the work on their own has kind of been really nice, because now they can just kind of come in the lab when they need to, and I don't have to babysit them quite so much. So they're getting a very hands-on, very targeted type of experience in that class, which, I don't think a lot of students get that opportunity, especially with this semester being, a lot of kids being online. Now we have also added some simulation stuff. Labster has been really good to us to kind of cut us some breaks on using a few of their Labster programs. And so a lot of the topics in our Cell Molec biochem class basically were introduced by doing a simulation, and then they actually came into the lab. So they learned the theory outside of class, come in and could actually just do the hands-on stuff. And it seems to be working. They seem to really be liking how it's working out. And again, I have a lot of data to kind of look at the very end here to see if this is a way that we may want to continue, at least with this particular class, is to treat it like what a graduate program would look like. Because that's one thing we just aren't very lucky here at USAO. There's no graduate programs. There's no access to graduate labs. I mean, we all have our little bitty tiny pieces of research, but not, we're not an R1 school. And so this gives them that kind of another level of understanding what graduate school is. Because I think a lot of them know what medical school is or PA school. And I think this gives them a chance to go, that's what you do, this kind of stuff. And I'm just like, yeah, this is what we do. And so I'm hoping that that becomes the model for this particular class, and maybe hopefully some of our other upper level classes, because this is actually a 4,000 level laboratory experience. And I'm hoping that I can continue to make it work. Even if we don't have to worry about COVID. I think this idea of giving them very hands-on, very directed experiences. And right now we're using a bunch of kits and stuff, but like from Biorad, so we can do CRISPR if we have time. Again, I think it gives them that opportunity to see what all the steps are, as opposed to just like this little piece that they may not understand. And so hopefully in the future, this will continue in this kind of a way. And it kind of just really fits in because it gives us a chance to talk about other areas where this may work. And with the combination of biology students and chemistry, so we have biology majors and chemistry majors in this program, it gives them a chance, specifically our chem kids, to see biology applications, because the chem program is pretty much all chem all the time. And they miss out, I think, on some of, well, why is this important? Because they don't have to do anatomy, they don't have to do physiology and those kinds of classes. So it kind of helps them make a little more sense of how to incorporate multiple disciplines into the same type of experience that they would have if they were just doing a biochem lab without any emphasis, I can't even talk, emphasize, whatever that word is, it's been a long day, to help them understand that there is a connection between the two. And that's about all I have to say right now. That hopefully there'll be more good information coming out of this idea that just sort of happened by accident on a Friday afternoon.
Well, thank you very much for explaining how what you're doing now came to be, and a little bit of the insides or the guts of that. Mark Walvert has posted the links to Labster and some linked regarding CRISPR there. But one of the things that just selfishly I'm curious about, and Dr. French had mentioned also the fact that one of the things that scientists are great at doing, even teaching scientists, is they look at the data in terms of student performance at the end of each term. And certainly that yields very, very interesting and usable information. But I'm wondering, now that you're 10, 11 weeks or whatever it might be into the semester with this new approach, from the teacher's perspective, do you have a sense that compared to students when they were only engaged in the way that you had it set up before this semester, do you notice student differences in enthusiasm and in attendance? Just from the teaching perspective, what has interested you?
I've been really surprised. The kids have all been, even if I'm a little bit late, like on posting what the lab looks like and stuff, they all come in pretty much ready to go. So even if I only give them 24 to 36 hours to process what the lab will be about, they seem to be ready to go, which I think is amazing. I do have one student who doesn't show up, but to be honest, she doesn't surprise me. She starts really strong every semester and then just, as you know, we all, like he said, we all have those students, that are like that. I think that their ability to talk about science is improving. We started out where it's like, oh, and then I took 5 microliters of this and I added it to 6 microliters of that. And now they're actually talking more about what that science means as opposed to, reciting the recipe. So I think they're kind of getting a point of understanding that. So they're going to have one big paper that they have to write over whichever lab they really feel the most comfortable with. So they'll write like a manuscript-y kind of a situation. And then they'll write what we call executive summaries, which is like a 1-page, these are my results, this is what it means, this is what we should do next, to give them that opportunity to work on their writing skills. Because that is one of the big things too that I think we need to work with our students, is not so much not just the talking about science, but writing about science in a way that scientists write. And it's one of the biggest struggles. And we even have a scientific writing class that they all take as sophomores, and we still have to remind them, no adjectives, no personal pronouns, no, this is not, you're not trying to impress your teacher with the use of big words that you may or may not know the meaning of. And so we start out in our ideas of this, is we kind of start out and we grade them pretty harshly, but they get perfect scores. So we give them lots and lots of feedback. And then as they move through, they get less help, and the grades are going to be penalized by, if they're if they are continuing to make the same mistakes over and over, because most kids just, they don't understand that, you know, the method isn't really the method. Everybody's method is based on somebody else's method, you know, and you can just go ahead and say, you know, based on, you know, it's such and such laboratory, whatever. And so I continue to struggle with some of those things, even though I've had these kids 2 or 3 times, and you're just like, I told you not to use that word. Well, it's statistically significant. Where's your stats then? You know, so like all these little pieces that you kind of have to bring in, and, you know, kind of, we like to talk about beating the humanity out of the student, so that, you know, that we don't, this is not your English paper. This is not, you know, British lit paper. This is a scientific thing. And we don't have time to read a lot of fluffy stuff. So just give us the content of what we need, and we will go from there. And, I think it's working. I'll be anxious to see if I can convince them all to do their course evaluations. You know, that's the other thing that's, I think, a hard thing to get kids to do. They're so tired by the end of the semester that the last thing they want to do is sit down and say anything. And they're like, but we like it. Yeah, I need that on paper. My boss needs to read that. His boss needs to read that. The president of the university needs to read that. Oh, well, maybe I'll get it done this weekend. I'm just like, oh. So, you know, it's just, kids are wonderful things. I love working with the students, even though some days I just want to pull all my hairs out, you know, but they're coming along. And I think, you know, I think this group is really going to benefit from how we've done this. And so we shall see.
Yes. Wonderful. Dr. French, I'm curious about this idea. Dr. Louch has just talked about students understanding how to communicate about science. And I'm wondering if there might be any effects of students working in this new kind of environment where maybe because the environment demands more written communication to communicate their ideas and their understandings, whether maybe there's a potential that there could be more of that kind of development in students becoming more facile more quickly in communicating science. Oh, you.
So, of, you know, so of course, a lot of the communication though that we're doing is live back and forth communication. So if you watch chats, yeah, communication goes up, but the format is actually something more comfortable with, which is the short, quick responses. Mm-hmm. In terms of other work though, so we've been having them write paper, write journal, essentially a journal article, a manuscript they've submitted for a while. So they're used to doing that. What is interesting, of course, for some of it is their ability to communicate what needs to be done, and realizing that people don't always understand what needs to be done unless you're clear about explaining it or asking the right questions. When someone says, I did this, well, what exactly did you do? And so many of the conversations seem to sound very much like us, which is, we might say when a student says, well, I did this, I heard you about significance or whatever. Well, where's the data? We need to have that for our paper, or where's this? So they're more cognizant in a different way, and they're communicating. And again, it depends. We're still working out, you know, in 5 or 6 more years of doing this, we'll get really good at it. But the idea of, you know, what partnerships look like for communicating. And I often tell the students when I'm talking to them that, you know, I'm listening and watching how my sons have to work now. And both of them are working for online, you know, for the companies they work for. And everything is this sort of combination of chatting and video conferencing and various other, you know, whether it's Slack or something else, a constant stream of information. And they should get used to this. This is, I think, some of the greatest experience they've ever had in real communication between each other, yeah, that they will have to carry on. And but that'll take a while. That'll take a while. It's interesting too. I think we sometimes forget, and this is not, I admit it, that this is not something I do with my students, but there are other forms of communication in terms of, yes, written, but communicating to a more broader audience, a public that might be different. And I think that depends, on what the goal of that, again, that goal of that course, what's the objective. So, so for example, and Jeanette, I can learn from you. I'm a behavioral biologist trying to get my students do molecular work, which, by the way, has served very nicely because I get to truthfully tell them that I'm totally ignorant of what they're doing. And so they should be free to practice communicating. And I'm going to ask ignorant questions because I don't know how this works. Now, unfortunately, as I've learned from my students over the last few years, I'm getting more proficient with molecular. So I'm not quite as ignorant. But, it's interesting when they have to say, oh, Dr. Fred, don't you understand how this works? Let me explain it to you. And that actually works very nicely, especially the first couple of times we did this. But there's also communicating to get into that broader audience. Someone said, well, what happens? So there's a classic piece of work in DNA barcoding where people go out and look at, one of the nice papers published a number of years ago was students were going out and looking at what's in the fish that's being sold in fish markets and in restaurants. Is it actually, you're familiar, Jeanette, is it actually the fish you say it is? Now, if you think about that, then you can convert that to an informational poster. You can convert that to something to explain to local newspapers, something you could communicate back to restaurants if they said, did I really buy what I thought I did? So there are other ways, and going back to the question, what makes something authentic? That's something that makes it authentic. So there's other forms of communication. One of my colleagues, Wouter Hoff, has been teaching an online upper division science communication, and they try to do a variety of different venues for teaching the students how to write for different audiences. And so I think there's a lot of value to that as well. Again, I can only point to other people and the excellent work they do. It is not something that I've tried to do. So we've just tried to mostly emulate the whole challenge of the peer-reviewed experience, where our students learn very early on to hate the third reviewer, because while the first two reviews were always good,
Right.
you will hear the same expletives many times coming out of their mouths. So we know it's authentic, because the students hate that third reviewer. But we explained it, and why is that third? What didn't they understand? Well, we have to learn how to communicate.
Right.
So that's part of it. Authenticity is very interesting.
Yeah. One of the things that I do with my non-science kids in a class is we do infographics where they have to, partly because it's everybody's in that class. So it gives the art kids and some of the creative drama kids a way to express science information in a way that's easier for them. And so we just finished up identifying whether some food products were actually GMO or non-GMO. We have to do some work on that again, because I think the mechanism that we did to isolate the DNA did not work like it was supposed to, because everything only came up positive in the positive control, and none of the plant primers worked, which they always work. So I'm not sure what exactly happened with that particular process, but we're working on trying to figure that one out. But that would be a good one that you could use as an infographic, as opposed to like a full science paper, or even as a scientific, executive summary, and so those are some of those types of ways to bring that science communication into multiple level courses. Now, Mark has a question about the impact on TAs and the stress from this new setup. Are you talking about what I talked about, Mark?
I think that may be a question for Dr. French, unless you have TAs without.
No, that'd be me. I am the TA and she is tired. Because my labs are actually back to back. So I teach micro from 12:30 to 2:30, and then I teach the cell molec technique from 3 to 6. And so, the TA at USAO is tired.
Yes.
So I'm happy to. So first of all, Jeanette, thank you. my undergraduate TAs, they come back and they volunteer for DNA barcoding. They actually enjoy the course so much, they come back and it's really being run by students teaching the next generation. But I will be happy to pass along, since they have struggled, that people far more professional than with much experience than I also, you know, sometimes the magic works and sometimes it doesn't. And they have learned that, and they pass that on with great authority to the next generation, that, you know, we're going to fail a lot. And, you know, the faculty here understand that, because it's real science, and we're very proud of them. So they'll be happy to know your students have.
I still do dishes, you know, so we've gone through 5 cases of 500 Petri dishes this semester. And that's just our solid media. So I still do dishes. So
yeah, we understand.
I don't really want to do dishes anymore. I'm just like, you are work study. I do dishes. You do dishes. When I can stop doing dishes, you can stop doing dishes. You know, it's that kind of a, I think that's part of why they can experience this, is because I make them do the lab tech part as part of their training. You know, they have to make media. They have to clean 72 test tubes to my specifications. And so, but yeah.
Well, let me answer Mark's question, if that's okay. And then I depending on time permitting or whatever, I've got a story. Not everybody washes dishes, and you and I, that's what happens, but it's worth mentioning. Anyway, okay, so impact on TAs. I think it depends. I mean, like everything else, I can't say there's something universal. I think that there are TAs that are exhausted, just like everybody else. But that's true during the normal semester. I think that I really don't think I have a good answer to say that COVID is anything more stressful. I think some of the TAs would prefer to work directly with the students. Others have learned to do our technique here and happily push the students away, not push the students, but do everything remotely. So I think giving them some freedom. I think I need to talk to them some more, to be honest with you, and find out more about what level they're at for some of this. I do have to again tell you that in the spring, and maybe it was that we all have to do this together. I mean, we sat down the night before we had to break to the first of these labs, or a couple days before, and the TAs just sat for hours. We were trying to figure out how to get Google to work for us, how to get everything to work for us. And they were very excited, not excited, but very positive. I'm not sure if it goes on for a couple more years, how they'll feel, but maybe we'll get better and it becomes more routine. Because again, we have TAs who, well, for a variety of reasons. So I can't really answer that in a good question. In a good way in particular about that. I think most of them just, yeah, I'll leave it at that.
Great. Well, realizing our time, maybe you can wrap us up with your story, Dr. French. You had said you might have one for us, time depending, and it will be a wonderful way to conclude the evening's event.
Well, I'm not sure how it's not a long story. It's sort of a quick comment, but you're right, Jeanette, we all have to do our work in the lab. And most of us, again, it's inspirational for students to see you side by side doing the same stuff they have to do. And I think that is important. And a lot of us, again, if people study, look at change over time with faculty, we start out young, hands-on with the students, and often students at, faculty at large research universities get further and further away. But a colleague of mine went to work at the Mayo Clinic in doing the genetics research. And when she was there, when they're hiring her and she got this, she was out there for the Mayo Clinic in a big research lab. You know what the reputation is. And they looked at her and they said, now understand, you wash no dishes. You just put everything into the sink, and magically at night, the elves come in and you come in the next day and everything is put away however way I want it, because that's what we do. We don't have time. You don't have time, given what we expect you to do, for you to wash any dishes. It's interesting, after a number of years working there, talk about, she went back to a smaller undergraduate institution and decided that was not who she wanted to be. But it's just very interesting. So when we think to ourselves, You know, this is part of it. Everything is always different someplace. So I don't know if that's a good story or bad, but I know that most of us sat there in just disbelief when I tell that story going, seriously? That's how, yep, we don't have time for that. You just put it in the sink. It all magically appears in the morning.
Wow.
And that's a very strange way to do things, at least for those of us who work with undergraduates here.
Yeah.
Anyway, Mark, I didn't know if you wanted to put up, it's on the end of my slides, but did you want to put something perhaps about ABLE and the forum on biolabs? That might be a nice thing for those who are interested specifically in biolabs. We had Merlot up there, that was nice, but ableweb.org, there's a forum on biolabs online, and that might also be a place where people might be interested. There we go. And there's a specific segment there, has been for biolabs online. People might want to chat and find out what other people are doing. Because again, we're all, yeah, that's it. Everybody is nationally. It's been very interesting. NABT also on their Facebook page is a constant chatter back and forth. How are we going to do this? How's that going to happen? So there's lots of people out there trying to reduce everybody else's stress over, oh my gosh, you know, we had this all planned out, and then, oh yeah, the power lines hit and this one happened, and then whatever we wanted didn't order. What can I do in the morning? And there's some very interesting solutions out there. So I hope that's helpful as well. That's at least what I can contribute for the night. And,
Thank you. Thank you very much, both to Dr. French and Dr. Louch. This has been very illuminating, and I think we achieved exactly what we wanted, which was an informal conversation, people leaving with good ideas, and a little bit of de-stressing and kicking back and enjoying collegial conversation with peers across the state. So thanks very much to both of our presenters, and thanks to Mark Walvard, who was our chat meister tonight. And we appreciate all of those of you in attendance, and we certainly hope that your power does not go out and that you survive the ice storm. So with that, we bid everyone a very fond good evening.
All right, good night now.
Good night.